disease within the IC, the International Group of Genetic Investigation of Anthropometric Traits, the GAIT Consortium, and GOLD, the Genetics and Lipid Disorders. Currently, our group works in identification and characterization of genetic variants in the Indian population, primarily in the Genome India project, and she is also involved in identifying the shared genetic etiology as well as causal interrelationships between metabolic risk factors and cognitive function affecting dementia. So the DBT New Delhi started the Genome India project just before the COVID, and it was from the Prime Minister's office that was initiated that nearly 10,000 Indian population would be sequenced across the major races of India. And her center is one of the core center where the entire project was evolved, and the project was initiated, and they coordinated with nearly more than 20 institutions across the countries, the universities, research institutes, and also the various organizations throughout the country to collect the 20,000 samples, and 10,000 samples have been already sequenced. Just last month, the minister had disclosed it in New Delhi as well. And so a lot of credit in the Genome India project goes to the Center for Brain Research, and Dr. Bratiti Kaheli was one of the key players in the center where, we have done this. So this is a very big achievement. So this is the first time the Indian population have been sequenced. And from Mizoram also, we have included around 148 samples, wherein more than 100 samples have been sequenced. And not only in Mizoram, across Manipur and other northeastern states, including Assam, and also the major races across the country. So her talk is going to be very interesting, and is going to have a very lot of translational applications as well. Yeah, please.
Good afternoon, everyone. Thank you, Dr. Senthil, for the really kind introduction. I must mention that Genome India is really team science. It's a textbook example of team science. So we just happen to be the coordinators, and collaborators like Dr. Senthil help us a lot. Today I will deviate a little bit from the Genome India work. We work extensively on Indian populations, but I'm also interested in understanding the genetic basis of neurodegeneration, and because population level risk factors like type 2 diabetes, dyslipidemia, and such are known to be epidemiological precursors for cognitive impairment in dementia. So we focus on that, and my talk will actually go through some of the recent results from our group. So, as I was telling that metabolic syndrome is a manifestation of several factors, right? So, for example, mostly abdominal adiposity, and as well as general obesity, and then dyslipidemia and hyperglycemia contributing to insulin resistance, and then hypertension could lead to damaging changes in our vasculature, and that can be related to impairment in cognition as measured through the cognitive scores that are done on study subjects. And we also lead studies for particularly these factors. So one doesn't really think that our cognitive function could be influenced by our genetics, right? We think or we not think, but the thing is that there are several studies where we have seen that the heritability can range from something as low as 22% to something as high as 51%. So when I say that, what I mean is that if you take inter-individual differences in cognitive measures, for example, if I can build a measure of general cognition, okay, and I have thousands of individuals, so the difference that I find in that particular phenotype of cognitive measure across all these individuals, how much of that variation in the phenotypic measure is actually influenced by the genetic component. So that is basically a measure of heritability. Okay, so the contribution from our genetic makeup in determining the variations for the phenotypic measures. So that's the heritability, where we are saying that it ranges from somewhere from 20% to 51%, depending on different studies and the psychometric properties of the cognitive scores that we are measuring. Okay? If there are quick questions, I can take in between also, not a problem. So now we did a couple of studies. The first one, we focused on British ancestry European individuals from this very rich resource, which is called the UK Biobank. Has any of you heard of the study, the UK Biobank? Maybe some of you have heard. So it's an extremely large biomedical resource where participants undergo, and it's 500,000 individuals enrolled over certain period of time, and they even have longitudinal measures, and they have an extensive range of clinical tests performed on them, and these are not any critically ill patients. Okay, so this is from the regular British health system. So what we did, as you might imagine, is that we take the cohort, which is about 200,000 individuals. We undertake a lot of quality control measures. If any of you are interested, I can go through them in detail. But this looks like a methodological slide, and it is a methodological slide. I intend to keep this for this kind of seminars where students are the primary audience, because we should really know that it is always good to be paranoid about your data. Always be paranoid. You really, really need to clean the data. Otherwise, it will throw us all sorts of spurious correlations and results, and we start believing them. It's really not good for us or the science, right? So we really undertake quality control, and then we control for the glycemic and the dyslipidemic variables. Our primary phenotypes are the cognition phenotypes. The 6 tests that correspond to the 6 different cognitive domains are, so we are not measuring general cognition. We are taking it in a domain-specific manner. For example, there is something which is called the simple processing speed, but there is also something which is called the complex processing speed. And these two, although they have certain overlap in brain anatomical regions, they are not exactly the same in terms of psychometrics or how these are measured and what implications they have. And more importantly, which one is more distorted or in dementia. There is a great level of heterogeneity at individuals' level in the population. And there are others like episodic memory, fluid intelligence, working memory, visual attention, etc. So we do conduct single variant association tests, and as well as we do club the single variants together to do the gene-based tests. But then it is also important to understand that it is a system functioning, right? So it's not like one particular variant will cause a great deal of upheaval and you measure that and you are done. It is not a Mendelian disorder. So these are complex phenotypes. Okay, so this is probably a right time to say that if you are measuring cognitive measures or type 2 diabetes or triglyceride levels, these are all complex phenotypes, which means that there are multiple genetic locations in our genome that have little effects towards the actual phenotype. And when I say heritability, I measure all of them together and I tell you the cumulative effect from all of these loci. Apart from that, there are environmental factors also. And then there is the complex interplay of genetics and environment. As well as there can be non-additive effects from multiple genetic loci that could be interacting. Okay, that is something which we call epistasis. It is not very well known or well documented for human populations because it is extremely hard to detect if we do not have extremely large sample size. But epistasis is something I'm sure or you coming from botany or zoology background, you know, right? I mean, it's really, really well documented in model organisms like C. elegans or yeast or Drosophila. And then we also try to understand what is the pleiotropic correlations between the metabolic phenotypes and the cognitive phenotypes. And then we proceed for other functional analysis and expression, how these genetic loci can influence the expression pattern in different brain tissues, or if they have ubiquitous expression across all the tissues. So what I did not tell here is that we are actually undertaking an association study, right? So essentially what we are measuring is the contribution from one of the alleles in each of the locus. For example, if you have an AC polymorphism, you know polymorphism, right? So you have an AC polymorphism. Let's say that C is the allele that will increase fasting blood sugar levels. So how much does one copy of C increase? How much does two copies of C increase? Is there any effect from AA at all? Because there will be some individuals who will be AA homozygous, right? So obviously, if it is an additive effect, you can understand that two copies of C will have the highest effect on increasing fasting blood sugar levels, right? So that is what is measured in a concerted manner in a genome-wide association study, because we are undertaking this particular test over hundreds of thousands of loci and tens of thousands or hundreds of thousands of individuals. Okay? So, what are the cognitive tests that are analyzed? Please feel free to tell me if I'm running slow. So, we have something which is called the fluid intelligence measure, which is the number of correct answers. So if the participant is asked 13 questions, and remember, these are not demented individuals. Of course, these tests are also done for people who may be in mild cognitive impairment stage, early dementia, through dementia, but these are tests which are regularly done on healthy population. Or what is the maximum number of digits remembered? So which is an working memory. For example, if I am shown hundreds of, not hundreds, maybe a dozen numbers, 10, 200, 5,336, 1,026. So what is the largest number that I can remember, right? Because it gets more complex. So that is your working memory. And then the complex processing speed is something how the symbols correctly match to the digits. So the participant is shown a matching pair of symbol and digits. And then after some time, he or she is asked, can you remember what this corresponds to? What this digit corresponds to? So in this manner, we also have, so if you can match the positions of the cards, which is the episodic memory, and then the time taken to press the buzzer for simple processing speed. It is also called the mean reaction time. This is one of the tests routinely done in memory clinics or cohort studies. And it's a very good measure of, it contributes, if you take weightage, it contributes a lot to the overall cognitive function. And then there is something which is called the alphanumeric trail duration, which is a measure of the visual attention, where a participant has to trace the path which comprises of alphabets as well as numbers. This is also very similar to something which is measuring the executive functioning. Okay. So then what are the results that we find? Oh, is this a little pixelated? It was okay on my screen. I'm very sorry about that. So here what we have is that maybe I'll try to give you an example from this one, because probably this is a little visible. So if you take, so this is a schema, it is not drawn to scale. So if you take simple processing speed, and if we do all these association results, what do we find? Suppose you have the baseline measurements, which means that you have not accounted for the lipid factors or the glycemic factors. Then you can have accounting for different lipid factors one by one in a staggered manner. Then you can have accounting for glycemic factors one by one, again in a staggered manner. What you find, interestingly, we found is that the loci that comes up as significant are actually different. Sometimes they overlap. Many times they do not overlap. When they overlap, we see it is probably something which is in the same pathway, right? So, for example, if we are trying to assess fasting blood sugar or HbA1c, and suddenly from triglyceride levels we have the same hit, we probably see that that is probably alluding to something like insulin resistance, okay? So these were a number of hits that we got. Usually when we say significant association results, our common parlance is to say hits. It's not a bad hit, it's a good hit. So here we are depicting the epistasis results. So for episodic memory, simple processing speed, and visual attention, we got significant gene-gene interactions that could be contributing to the phenotype in addition to the single variant results. Okay. So what we find is that there are 15 loci which were pleiotropic across metabolic and cognition phenotypes. There are also 4 loci suggestive of mediation via any of these phenotypes. What does that mean? It means that suppose ATP2A1 could affect one of the cognitive domains, right? But when you control for, let's say, serum glucose levels or serum HDL levels, the contribution is still significant. So, first of all, it is related to cognition. Secondly, it is probably mediated through the HDL levels or the serum glucose levels, and the effect is diminished, but the effect still remains significant. Okay? So this was actually a great motivation for us to undertake proper causal analysis in the next study. And the genes that you cannot read here, again, I'm very sorry for being pixelated this diagram. So what we find is that these are the genes that are associated or enriched in these kinds of pathways, which are long-term depression and something that is involved in the activation of protein kinase activity, etc. So lots of synaptic genes as well as neurotransmission processes, which actually tells us that this is how cognition will work, right? Neurotransmission, synaptic plasticity, and all that. And that is what we are uncovering from studying general cognition. So our idea, the point where we started, is the neurogenetics to understand cognition at the general population level in order to know the disease process. If we don't know the genetic mechanisms of underlying our regular cognition, how do we know how cognitive impairment works in dementia, right? We first need to understand some kind of a reference. So I was interested in that, and the student was spectacular in doing all this. So we identify the cognition associated variants, right? Do they mean anything for other brain morphological traits or other metabolic risk factors? Because metabolic risk factors, as you can see, is a common theme that is one of our interests. So what we find here, so these were 18 plus. Mostly healthy individuals. We took out the dementia individuals from this study. 18 plus, and the average age was somewhere in the mid-40s, and the highest age was probably 80. So what we also find is that these loci are implicated in something, the white matter microstructure. So white matter hyperintensity is one of the things that you notice for early dementia, right? So that actually tells that these cognitive scores are pretty non-invasive measures, and when we are quantifying these, in future we can have certain gene sets that would be useful in a population-specific manner to see the genotypes and tell that who could be at risk, right, in the population. So the brown ones were for correlating with brain-related measures, and the blue ones are the metabolic risk factors. You can see here pulse pressure, pro-insulin levels, and insulin-related traits and all that, BMI. So there was very clear distinction of 2 types of loci that are implicated in cognition. one that is affecting brain microvasculature or vasculature, and the other set which is hinting us at metabolic risks. So this was again a great motivation to test for horizontal pleiotropy because it is impacting brain structure traits and metabolic phenotypes. So what was the biological relevance, right? So this is a summary of what are the genes that we got in 6 different domains. So they are color coded, right? Overall, all of these genes link to synaptic plasticity, connectivity, neuroinflammation, and oxidative stress. Everything that we know are disrupted to one or other extent in neurodegeneration. So this is again the overall results. But what I would like to tell you about this thing is that the dark red ones or the brown ones, whichever way you view it, these are the genetic loci which can serve as expression quantitative risk loci for expression in relevant brain regions. For example, if I'm studying executive functioning, one of these genes is probably having altered expression or can control expression in the prefrontal cortex. So that means a lot, right? That is definitely one of the more relevant hits than the other not so relevant ones. There can be EQTLs for nearby genes, which is extremely important from a genetics point of view, is because we do not always have the statistical power to pinpoint what is the causal gene. And that is the normal scenario. So we do not miss out on annotating the nearby genes. And many of these loci do influence the expression level of nearby genes. There can be EQTLs for both the annotated and the nearby gene. In these cases, it is extremely pertinent that we try to tease out what is the actual effect and if there are any interacting effects or not. Then there can be gene-based hits, which is from the bold ones. So this was very encouraging for the student because she was like, now we got LRP1. So why? Because LRP1 is implicated in the pathway very directly for A-beta cleavage, degradation, clearance, deposition. But, you know, genomics is one of the areas from where you would want an evidence to go for further drug development in today's world. And LRP1 was never coming up. Somehow we got it studying this particular population and focusing on the general cognition in the general population. So this was very important, which means that it is probably lurking there. We just need to look closer and deeper to understand what variants in LRP1 could be responsible and if that could hint us at more of the disease etiologies and pathways for neurodegeneration and specifically Alzheimer's disease. So this I already told, the suggestive. So everything that we are seeing, disease related or not disease related, right? It should be considered in the bedrock of evolution. Okay, so what is the evolutionary constraint of the associated variants? What we see here is that it's not always the norm that negative selection works and the variants are purged out. It is the norm, but there are deviations also. And so what we see, these are color coded according to the allele frequency, which means that how prevalent they are. And what is the percentage of variants explained, which means how much they influence the trait. Remember the first slide. So what we are seeing here is that if they are more intolerant to functional changes, which is on the, so the low score on this side means that they are more intolerant. So they should have been purged out, but they still remain. But that doesn't mean that all of them are rare. Some of them are quite commonly present because you see, they are greater than 5% in the population, right? It's not like they are 0.1% in the population. This is something which we are also seeing from our Genome India data, that we know that something is very harmful, so it should not be present. But we see that such variants are present, maybe not in huge numbers, but quite commonly present in the population. And so these actually tell that you can stratify the study population in certain pockets where you can find protective and risk factors to specific disease conditions. Right? So what did I just say? That there can be both rare and common disease-associated variants that are so-called intolerant to functional changes, but they are present. They are contributing to the disease. Doesn't mean that all disease-causing variations are rare. Maybe for monogenic disorders, but not always for complex phenotypes. So I'll skip this because this is a summary slide in case someone was interested what is the take home from this particular one, where we took the interactions with ApoE. So we have evidence of pleiotropy, mediation, and several risk factors. So okay, I have gone through this, so I'll skip it in the interest of time. So our next study goes deeper into more neurocognitive outcomes. And this is done on a rural cohort in southern India. So these are similar methods. I already explained them. I'll not go into the details. I just want to highlight that when we are doing array typing, you know, people who work in the genetics labs and zoology labs, that you are doing only a few hundreds of thousands markers, right? But they are not always informative about the whole genome. But it is also prohibitively costly and analytically very tedious to do whole genome sequencing for everything you have. Why would you do that? And more than everything, why should anyone give us the money for doing every individual sequencing, right? So what we developed is that an in-house haplotype reference panel. We did it from one of the Indian studies, not Genome India, although we have done it for Genome India also. But here I present nothing from Genome India. So we did it merging the South Asian known haplotypes and the ones that we constructed from our own study. There is no overlap of study individuals in both the South Indian studies. And then we construct this reference haplotype panel, and I'll just show you how that has given us the power that we can actually identify associations which would have been otherwise impossible with only 5,000 to 6,000 study sample, right? So these are the minor allele frequencies which we are imputing, and these two lines will tell you the worldwide used reference haplotype panels for genotype imputation. This is the a measure of the imputation accuracy in R square. So you can see here that for our panel, which is in green, so everything is close to 80% here. For something present very as low as 0.5% minor allele frequency, which is pretty low, right? So, but if you take the worldwide accepted standards, you would have to have something at around, at least you see here, yeah, at least at 2% in the population. Did I say it correct? Sorry. Yeah, 2%. 2% in the population to even detect any effects, which would be accepted by worldwide standards because imputation accuracy matters a lot. So this is another thing which is showing you that how much of the genome do we cover. So it basically tells us that at least 80% of the genome is covered, even if they are from low frequency variants, in understanding the contributing effects of the genetic variants. Yes, that would be okay. And these are the imputation accuracy for different types of alleles. So what do we have here? We have certain good hits. So this is a, if any of you are interested, so this is a quantile plot where you will have the observed p-values and the theoretical p-values. So if something is deviating from this 45-degree line, which is like over the random, right? So it is not random effect. So you can see that something is turning out to be significant. If you have a very good sample size, good phenotyping, good genotyping, you will see very nice peaks here. So coming out like this, you'll see the snake things. So what we find here is that there are novel hits which are genome-wide significant for visual attention in the Indian population. Similarly, we find in the memory domain where the test is about recollecting names of individuals. And see, this QQ plot is even nicer. So, and these are all rare variants, right? So, which tells us something that we are hitting something probably very unique to the population. Similarly, we have single locus association tests for the metabolic risk factors, and this particular population, as it turns out, has really high features of dyslipidemia, and which actually reflects in all the genome-wide association hits that we are getting from here. Okay. We also have auditory attention with at the subgenome wide level. So you can see that we are really underpowered here. And what I would like to show you is that when we see, so I chose snippets from each of the cognitive domains so that it doesn't become too overwhelming. Otherwise, it will be a 100-slide PPT. And so we have top 20 enriched pathways for executive function. You can again see from the UK Biobank as well as from the Indian study. So depression is always the major hit. Okay. And with the p-value, as you can see here, adjusted. So there are others like amyotrophic lateral sclerosis, cardiomyopathy. So there are some non-specific hits, but mostly dementia and brain phenotypes. For example, FTDs with motor neuron disorder or mental retardation. Okay? This was an extensive pathway enrichment analysis. This Indian population work is unpublished, so please understand that this is work in progress. Do not disseminate and do not learn for final from these slides also. Similarly, what are the other genes that are coming up? You can see that this entire region is focused on brain-specific genes. And you can see that APP or SERF2, like things that are known to be implicated in Alzheimer's disease, especially APP, right? Whoever thought that you will study general cognition in 5,000 individuals and you will get hits in APP for executive functioning. But that's the beauty of genetics. So I'll end with the causal analysis. What we are taking is that the dyslipidemic or glycemic risk factors, and we are trying to see if they can influence the outcome with lots of assumptions that should be validated first, because otherwise the causality answers do not hold true. That it should be only through the exposure, which is the lipids, and there should not be any reverse causality. So the outcome phenotype should not be related to these variants directly. Then your theory, your hypothesis that it goes through the exposure falls flat. Okay? So after all this, what we see is that, again, this is just one of the snippets. What we see is that there is a significant effect of memory domain on a parameter triglyceride HDL ratio, which is also a very good marker for insulin resistance, especially in the Indian population. And then these are not false positives, as you can see from the funnel plot here. The effect is very low, positive direction, so which it means that higher people with higher proportion of insulin resistance will take longer time to recall something for the questions. Okay? And this is highly significant. We could not say that individual variants had very significant effects, but pooled in a fixed effects manner, that was significant. There are other results. So I'll stop there. What are the key insights is that we have genome-wide signals for several domains of cognition, causal effects of dyslipidemic markers on attention and memory domains. And when we collapse the coding variants together, we hit very plausible pathways that could be responsible for executive functioning, visuospatial, and language functioning. Funding, and I always say that everything is possible because the study participants cooperated with us. Without them, we could do nothing. These results were shown from the SANSCOG study. Dr. Jonas in CBR takes care of day-to-day things for this study, and he is the PI. TLS study from which we built the imputation panel, Dr. Thomas in CBR, and these are my personal grants and the workhorses. They are the stars. Whatever we tell, they have done. So I think I should probably tell by name. Where is she? Yeah, Shreya has done most of the cognition and the metabolic risk factor work. I did not present anything from Kritika, but she's the workhorse for Genome India. So we would be blind without these two. And definitely the others are also catching up, the PhD students. Thank you so much. Okay.
Yeah. So, Dr. Prithvi, yeah, thank you for the excellent talk. It was very interesting. You have summarized it very nicely from the bioinformatics perspective, as well as gene environment interaction, and also the metabolic pathways. So, one simple point that if you could kindly highlight, like for example, in Genome India project as well, common variants as variants of diseases, right? So, like for the benefit of the students here, so sequencing patients and getting the biomarkers is excellent work, which you have also given a case example. But sequencing healthy individuals also as a to know the common variants in the population. Yeah, if you can kindly highlight.
It is very important because otherwise we will not know the population specific prevalence of variants. Because we might be thinking that something that is implicated, one particular variant that is implicated in, let's say, European population is implicated for disease context in ours also. And that could be entirely different, excuse me, because that common variant could be rare here and vice versa. And sometimes we have unique variants to our population in and even in an ethnicity specific manner. So we are seeing in Genome India, as you also know, that if you take groups of individuals belonging to certain ethnicities, you will see that there are a unique set of variants which is as of now not present in other ethnic populations or other world populations, right? And it is very common even in today's world when sequencing is being done rampantly by developed nations. You still sequence any South Asian population, you will probably hit somewhere between 6% to 12% unique markers. Of course, this number will keep slowly going down as we sequence more, because then we know more what kind of variants exist, right? So the novelty factor decreases, but that nowhere takes away from us that we need to sequence the healthy population to understand the reference in our population, and that would serve as a knowledge base for understanding the disease. Thank you.
Certainly, ma'am, a very good presentation and very good big data means 20,000 target and you have done 10,000. So with this huge amount of data, can you have any particular workflow so that from the whole exam sequence analysis to this kind of a, is there any kind of a stuff you have designed for the common people to use those your platforms to analyze?
Yeah, yeah. So that is what I was presenting as details. I mean, anyone who wants to learn is always welcome. But yeah, we have a very standardized workflow for starting from genotypes, taking care of the phenotypes, and then how to do the association with appropriate quality checks in place. And how do you understand what the associations mean for us? Yes, there are standard
Is
workflows.
it available online?
Yes, that's also available.
Yeah.
Actually, the GitHub page of the published paper will give. It's very detailed.
Okay, thank you, madam.
One question is there. Okay, so if we know that, so okay, so it it can be done. It can be done. So there have been examples because Indian population has been grossly understudied. So I have no choice, but I can take examples from other populations. So European ancestry individuals are the most sequenced and the most studied, conveniently, right? And it's good also because we have developed our preliminary knowledge base and we are still developing by from those studies. But at the same time, it has been found that there can be variants which are implicated, let's say, for type 2 diabetes. They were never found in the European population because those individuals have because the population has very rare prevalence of those variants. Okay. But those, sorry, I'm coming to that. So, in the general population, right? Now, so when it was sequenced in, it was not targeted, the particular individuals, people did not find them. Then they went to East Asians. So in certain East Asian studies or African American studies, it is coming up the prevalence of variants which are related to type 2 diabetes, and they are not rare. They're quite common in the population. So when researchers go back and check, it has happened that highly diabetic individuals actually have these variants, which was earlier being kind of clouded because we were taking that as a whole. And in these kind of statistical genetics, we do need the numbers. So we cannot always focus on the tails of the distributions because that could lead to spurious correlations. And in that process also, what happens is that we might be missing something really rare. But on the other hand, even if we have that, we would not be able to show that their effective is significant. We have to go to another population and then facilitate the fine mapping of the disease loci.
Excuse me, sir. We are already running behind the schedule. Please, you may please discuss during the lunch time. You will be having enough time.
Thank you, everyone. Oh, okay.
Thank you, Dr. Bertati, for a wonderfully informative session. I now request Dr. Anil Kumar to present Dr. Bertati with a small token of appreciation. I request Dr. Syama sir as well.
I hope you all enjoyed our break. This is an announcement for the oral presenters. Mizoram Macro and Perveen are requested to submit their presentation. Now we shall move on to the final and last plenary session for this afternoon. To commence the session, I would like to introduce our session chair, Professor G. Subramanian, and our session co-chair, Dr. Vikas Kumar Roy, Department of Zoology, Mizoram University. I now hand over this session to our chairperson.
Good afternoon to one and all. I hope you had lunch. Now we are going to have food for our thought. So I am the chair, Professor G. Subramanian, head Department of Zoology, Mizoram University, and my co-chair, Dr. Vikas Kumar Roy, Department of Zoology, Mizoram University. So we are going to conduct this particular session. And in this session, the invited speaker, Professor Jogan Chandra Kalita, he is the head, Dean and Head, Department of Zoology, Guwahati University, Guwahati. So with due permission, I would like to introduce the speaker and his accomplishments. Professor Jogan Chandra Kalita is the Dean as well as the Dean of Faculty Science and Head of the Department of Zoology, Guwahati University. And he has awarded prestigious Commonwealth Fellowship in the year 1995. And apart from that, he's also having prestigious Lever Hume Trust UK Research Associateship for postdoctoral research in the year 1999 to work at King's College, London. And he obtained a PhD from King's College, London. And apart from that, he got INSA Royal Society London Visiting Research Fellowship in the year 2006 to pursue second time postdoctoral work, again at King's College London in 2010 to 11. And apart from that, he acted as a director, College Developmental Council, Govt University, and director UGC HRDC, Govt University. Professor Joganchandra Kalita visited many countries like China, Japan, UK, Belgium, Bhutan, Bangladesh, and Vietnam. And he has authored 15 books, published more than 150 research publications in the peer-reviewed journals, and produced 27 PhD research scholars. And also he is having one patent awarded in the year 2020. And he is also a member of so many academic bodies and member of College of NAC Monitoring Committee also. And he has successfully completed 7 major research projects from various funding agencies like UGC, DST, DBT, Ministry of Environment and Forests, and so on. And with this introduction, now I call upon the speaker, Professor Joganchandra Kalita. Yes, time will be 30 minutes. Yes. Okay. This is the thing, sir. Yeah.
So, good afternoon, everybody. Okay. Honorable Chairperson, Co-Chairperson, esteemed professors sitting here, all young friends coming from various parts of from Thailand, Professor Ramesh Jimbo Bunaratana, with whom I have connection for last more than 15 years. And he has connection with India for more than 50 years. He is a product of Assam or Punjab Agriculture University. Esteemed guests, all well-wishers and student friends, my time is 30 minutes and I will try to manage. Actually, my field is endocrine disruption. Last 40 years, I have been working on endocrine disruption. And the title here is the detection of hormonal activity. Hormones are nothing but the products of endocrine glands. So therefore, we can include some of the plants as phytoestrogens. So those plants, they have products like estrogens in us, and those products are termed as phytoestrogens. Phyto, plants, estrogens, hormone of our body, mostly from women. And when we talk about these chemicals, we find that there are some beneficial effects. At the same time, there are reports that there are negative effects, which are deleterious effect, adverse effect, and here I will try to show you one devastating kind of things happen that was reported in 1947 by the farmers. And I want to let you know that my talk will be a little bit general because time is less and I am addressing mainly the young friends, that what we can do from this part of India. Because our part of India is biodiversity hotspot. We have many resources, mainly some of them are endemic, and we need to explore what they contain in them. So therefore, that was the first thing happened that was 1947. People are not even thinking about the hormones. Those were termed as phytoestrogens later. But effect was like infertility developed in Australian sheep population. That was the largest part in the society. The economic growth depends on that. When economic growth depended on that kind of particularly farming, and when they were at a stake, that was reported and discussed in the parliament. You can minimize the light. Yeah, it may not be visible to the end. So one or two lights you can put it off. These two lights, yeah. So now it is better. So then when the farmers were affected, country was affected, that was discussed. And when the matter was discussed, scientists across the world, they were invited to do some kind of research to find out the culprit, why that had happened, why the Australian sheep all of a sudden got infertility in them. And sharp decline was the major findings. And later on, after 10, 12 years, it was in 1947, that was in 1962, first chemical discovered from the grass on which the sheep were fed. And the chemical was cumestrol. And it was found that that was like having estrogenic property. And Cambridge professors and many others, they worked together. They took more than 10 years. And today, you, the scientists, you need only 1 year to find out that. And there is the problem of actual techniques. These are called measurement techniques. I do use animal as well as in vitro techniques like cell culture to screen some of our plants of this part of India with traditional knowledge that they were used by the people in the past or maybe in the present by some practitioners, herbal practitioners, for treating infertility or some kind of diseases. And depending on their information, we work. And we work together, developing collaboration with many universities across the world. So how they look like? You can see here our estrogen, that is cyclopentanohydrophenitoin ring. That is the steroidal ring is there. But in all other chemicals, as I said, cumestrol, which was the culprit present in the grass on which Australian sheep fed, and later on it was found that infertility developed to maybe around 100%. So therefore, it was analyzed and it was found that many of them are not steroids, but they are steroid-like. Chemicals in plants, they are steroid-like 99.9%, like our own endogenous estrogen. When they are coming from outside, we term them as xenoestrogen, xenoestrogens, but they are not real estrogen. How then they work in us, within our body? They mimic. Suppose I am Jugan Kolita speaking here, somebody may come in my disguise, can deliver the lecture. And it will be very difficult if 99% similarity is there. So that way they mimic, they block the actual hormone action. Many of them were identified, still there are many to identify. And why to identify, why to detect? That is the most important agenda of today. Because we need to know the plants like resveratrol. Resveratrol was a plant chemical, a phytoestrogen present in grape juice, grape, the skin of the grape, red grape. And Cambridge professor, they found out that that was protective for our heart. Even the people, if they use, then our heart may be protected. That way, the beneficial side, I would highlight. First, I talk about the devastation, that was the bad effect, and there are plants where phytoestrogens are there, and they are our plants. And if we work more on them, even we can go to the development of drugs, so discovery of drugs. So therefore, bioinformatics, Professor Guru, he has helped us through bioinformatics in our university Department of Zoology to find out some of the chemicals very quickly. Otherwise, it will take a long time. So if you want to know about what are they, mainly for the young scientists, so these are the products. The names Zenistate, Digen, Cumestrol I talk about, Biosenine, and many. And many of them are having some good effects on us. So therefore there is our interest. I mean by our, that scientists. Many of the scientists, they wanted to know how many good estrogen we can find in nature. Like resveratrol was a good estrogen. Genistein, good estrogen. In some European countries you can find the label is there, a brand is there. Bread in the name of genistein. That this bread in the morning time we use in the breakfast, that contains that particularly phytoestrogen, and if you use it, that will help you. So your health will be protected from some kind of diseases which are termed as Western diseases. I think all of you know about Western disease. Western diseases are those diseases, those could be protected by Eastern food. Eastern food, who take Eastern food? We the Indians, the people of northeastern region of India, Japan and China. And what protection we can get? Protection from Western diseases like hormone-dependent cancer, heart disease, or many kind of hormone, or even the cancer as a whole. So major classes like this, I am not going to detail about that, but these are already identified. And as I have said, that many more are yet to come. We identified one, that was one new, and that was termed as 8-phenylnaringenin. 8-phenylnaringenin we identified from one plant, and that took me 3 years. So 3 years work, but today, I think young scientists, you can do and find out the actual chemical maybe in 1.5 year time, provided you use both in vivo, in vitro, and bioinformatics together. So phytoestrogen and beneficial effects, there are many. As I have mentioned some of them, you can Google and you know that every day, right from our breakfast till our dinner at night, or in many occasions the foods we use, we have good effects, and at the same time some of them may be having bad effects. So there is the major role of the scientists like us to characterize. And if we can characterize actually, then we can talk more about them.
Hi everyone, good day. Thanks so much for the invitation to speak to the International Conference on Current Trends in Biological Sciences. And thanks so much to Dr. Sandil Nachimuthu for the invitation to speak. Today, I'm very happy to tell you a little bit about the work that we're doing here in my research group. We are based at the International Laboratory for Human Genome Research, part of the National University of Mexico. And that is this yellow building here in the middle. The other places you see in this picture is just collaborations that we have for this project. So hopefully I'll tell you a little bit about genomics and transcriptomics in Latin America, and hopefully you'll find something interesting in this talk. Okay, so first a little outline of what I want to tell you about today. First, I'm going to tell you a little bit about melanoma, which is a type of cancer that we're interested in the lab, and how it looks like in Latin America. Then I'd like to tell you a little bit about our team's efforts to study melanoma, and with these We'll include some preliminary findings from whole exome and transcriptome sequencing of a subtype of melanoma called acral melanoma in patients from Mexico, and also tell you a little bit about how to build a collection of patient-derived xenograft models, which are in vivo models for cancer growth from these Latin American patients. And finally, some take-home message and acknowledgements. Okay, so let's start with the beginning about melanoma in Latin America. So first, what is melanoma? So melanoma is the most aggressive type of skin cancer because it is when it is diagnosed at an advanced stage, it has a poor response in general, and it causes the majority of cancer deaths from dermatological malignancies, as it constitutes only about 4% of all dermatological cancers, but causes about 75% of deaths from skin cancers. Now, its cell of origin is a melanocyte, so that's what is called melanoma. And in this figure here, you can see a 3D structure of the skin. Here at the top, the epidermis in the middle, and the hypodermis at the bottom. And if we do a zoom in here in this junction, or this frontier between the epidermis and epidermis, then we see different skin cells, and we see here that melanocytes actually live in this what is called the basal layer of the epidermis. And their main biological function is actually to produce melanin in response to UV radiation. So when we go out in the sun and we get a lot of UV light, then cells detect that, melanocytes start producing melanin and giving it to other cells in the skin. And melanin is actually able to absorb the most damaging components of UV light, and that way it can protect DNA from the cells in the skin. So their main job is to respond to UV damage, UV radiation, and prevent DNA damage. Okay, so the majority of melanoma cases are actually caused by an excess of this UV radiation because the sun, as we were discussing just now, can cause mutations in DNA. And if we get a lot of mutations from the source, then that can accumulate and that can lead to cancer. And about 90% of melanomas are caused by sun irradiation. UV radiation, sorry. Now, another interesting thing about this type of cancer is that the incidence has been increasing for the last 40 years. So if you look at this graph, this is based on numbers from the US, but this trend is actually repeated around many places in the world. You can see that from 1980 onwards, the incidence of melanoma has something like multiplied by 5, let's say, quintuplied. And that may be due to two different factors, or it's possibly due to a combination of these two factors. The first one is that we are now better at detecting melanoma at earlier stages because we know more and we have better equipment. So more melanomas at earlier stages are diagnosed, and that increases the number. But also that there has been a change in people's behavior in the last 40 or so years, in which little short holidays where we go and expose ourselves in the sun are actually much more common now, and people get more exposed to the sun. And in the end, that's also a factor for this sharp increase in melanoma. And it is thought that, as we were saying, it's a combination of these two factors. Also, it's important for us that melanoma, when detected at later stages and treated at later stages, is very highly refractive to chemotherapeutic treatment. And as you can see in these curves, patients very seldom respond to these treatments. This is important for us, at least here in Mexico and in many places in the developing world, because most patients are still treated with chemotherapy, as some other types of treatment are more expensive. So that's also a point of interest in our lab, why we study this type of cancer. And another interesting thing, and now talking about the genomic landscape, then we know that about 80% or a bit more than 85% of cutaneous melanomas, that is melanomas that arise in the head, for example, or the arms or the legs, have driver mutations. That means mutations that actually give a selective advantage to the tumor in these genes called BRAF, RAS, and NF1. And the students here on molecular biology may already know these genes because they are part of this very important signaling pathway called the MAP kinase signaling pathway. And this is a growth a pathway for the cells. So the mutations in these genes activate this pathway, and that means that those mutations actually confer a selective advantage to the cell in which it can grow more than its neighbors and eventually can form a tumor. Okay, so but melanoma, as with many different types of cancer, is also divided in subtypes. And in the case of melanoma, here I'm showing you the 6 major histopathological subtypes. So the first 3 here that you can see, superficial spreading, nodular melanoma, and lentigo maligna, are actually all caused by UV radiation. And they are more common in people of European descent and light skin individuals. And the 3 here to your right that you see here, mucosal, uveal, and acral, are actually rarer melanoma subtypes. They are not as common. And as you can see here, they show in other parts of the body. For example, mucosal, as its name says, is in the mucous parts of the body. Uveal is in the eyes, and acral is actually on the soles of the feet, the palms of the hands, and under the nails. And this is the type of cancer of melanoma that we're interested in our lab. Now let's talk a little bit about that and why we're interested in it. So as I was saying, it arises in the palms of the hands, in the soles of the feet, and under the nails or subungual locations. Now it is misdiagnosed as other conditions usually because it looks like, as you can see also in the picture, as other lesions, for example, fungal infections or diabetic foot. So in many cases, the patients get diagnosed as other things before actually being diagnosed as cancer. And that means that they are diagnosed at later stages, which we will see later. And finally, as we were saying, it's a rare subtype of melanoma in European descent individuals, as it comprises only 1 to 8% of the diagnosis. However, if we look at a map of the distribution of melanoma subtypes in some countries around the world, then we can see that this subtype acral that you can see here in red actually constitutes what we think is the majority of the diagnosis in countries such as Mexico, where I am based in, others in Latin America, such as Peru, and then some others in Africa and in Asia. So this is also why we're very interested in this subtype. And it is not only the most common type of melanoma here and in other countries in this continent, but also it is understudied. So if you just take a look at the clinical trials that have been done in melanoma, you can see more than 3,000. But if you focus on acral melanoma, then you can see that it's only about 41. And not only is acral melanoma understudied, but also the populations where it is more common that we discussed before, populations in Asia, in Africa, and Latin America, are also understudied. And in this part of the figure, you can see the distribution of ancestries of samples in different repositories. So here you can see 4 repositories. The first one is the pan-cancer analysis of whole genomes. The second one is the Cancer Genome Atlas, COSMIC, which is the catalog of somatic mutations in cancers, and the National Cancer Institute Patient-Derived Model Repository. And as you can see here, there's a lot of blue, which means European descent samples, and very few from other places. So all of these together just means that we're very interested in a subtype of cancer that is not very common in many places in the world, but here and in other countries where populations are understudied is the most common type of melanoma. And that is why we're interested in this cancer. Now, here in Latin America, just to make a little note, and probably some of you may also feel the same, that there are many challenges to doing genomic research in countries in Latin America and other low and middle income countries, and these are some of the reasons why. So first, there's not many studies of our populations available, so there's not a lot of let's say repositories to compare against. Also, there is usually a disconnect between the medical and scientific communities. Usually these communities do not have the same objectives, and it's difficult to work together, which also hinders the advance of the projects. And also, it's usually more expensive to carry out research because we have to import the reagents, and that get taxes and transport costs. So in the end, just to say that much of the work in our lab is to establish facilities and workflows, and also to train people to be able to do this analysis. Okay, so that was the introduction, and now I'd like to tell you a little bit about our work. And first, to tell you about some findings from whole exome and transcriptome sequencing of this type of melanoma in Mexican patients. So first, what did we do? So we started a collaboration with the National Cancer Institute here in the country, in Mexico, to start recruiting patients with this diagnosis, and we have been doing this since 2018. And once a patient goes with this diagnosis and accepts to form part of the study, then they donate a part of their tumor in the form of FFPE, which you can see here in these pictures, and also saliva, so we can get also the normal DNA from the person, and we can compare tumor versus DNA and understand the new mutations that the tumor gained. Then once we have that material, we do DNA and RNA extraction, and then we do sequencing of tumor normal pairs. So first, this is how our population looks like. So we have 128 samples, and this is from a total of 96 patients. Some patients have donated more than one sample. You know, sometimes they have primaries and metastases. And you can see here the majority of them were diagnosed in 2017, slightly more female patients than male. The majority coming from the center of the country, which makes sense because Mexico City is here, and that's where the hospital is, so that makes sense that patients that are close are more common. And as I was saying, these tumors get diagnosed late, so they come ulcerated and at stage 3 usually, with a lot of spreading ages from 32 years of age to 98, with a median of about 60. And these are thick tumors, and the majority, as you can see, come from the feet. And we have access also to some metastases that you can see here in blue. So if we first look at the genomes from these patients, then we can see that indeed these patients are mixed American. And this is important to check just because there's, like in India, a lot of different ancestors here in the country. We want to understand what ancestries we are getting in these patients. And here you can see the 1,000 Genomes Project. That's the first 5 components. The African ancestry component is in green, and you can see here each line is a different person. East Asian is in orange, European in blue, South Asian in red. And as you can see here, mixed Americans have a component of blue, which is the European component, and some green and some yellow, which is the Amerindian component. And as we can see here, our patients actually look very closely like the admixed Americans in the 1,000 genomes. So we're happy that these patients are actually of Amerindian ancestry, which is understudied, and we're happy to be able to study them. Okay, now this is an oncoplot, and this is what the results look like. This is the work of Patricia Basurto, who's a PhD student in our lab, and this is what you can see when you look at a tumor, a cancer's genomic study. Each of the columns here is a different tumor, and each of the rows is a different gene. So if you see a colored spot, that means that that tumor has a mutation in that gene, and that way you can read this graph. Okay, so here we can see that the majority of samples have mutations, not the majority of patients have mutations in these genes that I mentioned before, the MAP kinase genes. Actually, less than 40% of these tumors have these mutations. And if you remember before, I told you that 85% of cutaneous melanomas actually have these mutations. So they have a much lower mutation rate. The most mutated genes is NRAS, and then also BRAF and NF1 follow closely, and these are genes actually in the MAP kinase pathway, as we saw before, but they are mutated at a much lower frequency. Interestingly, KIT, here the second most mutated gene, is highly mutated in this population, and also it's a different gene that also activates this MAP kinase pathway. But that's a difference with cutaneous melanoma. And if we compare these against other studies that have been done on oropharyngeal melanoma in different populations, then we see that it looks similar as the genes mutated look similar, but also the frequencies can be quite different. And we are investigating whether that's because of the environment where the patients live, or it could be something genetic, or it's just random sampling. So that's something that we're investigating at the moment. Also, we can get a copy number landscape, and here what you're seeing is all the samples put together, all the chromosomes, and if you see red peaks, that's a gain, and if you see blue, it's a loss. And here you can see genes that are gained and genes that are losses. And what we can see is that known oncogenes are amplified, like cyclin D1 or CDK4, whereas tumor suppressors like CDK and 2A are commonly lost. And these also look similar to the profile of tumors from the Australian cohort, but some important differences are also seen where we don't see amplifications, for example, where they see amplifications. So we're also trying to investigate whether this is a real difference or it's also ascertainment bias. Okay, then we can look at the DNA copy number profile now by sample, which is what you're seeing here. Each sample is a column again, and each chromosome, it's a row. And red is a gain and blue is a loss. So here we can see that there's actually differences in this copy number profile depending on the mutational status of the samples. And tumors that have mutations in BRAF and NRAS actually have fewer of these chromosomal aberrations. And that may mean that they need fewer mutations or fewer variants to actually form a tumor. So they're very potent oncogenes. Also, tumors that are under the nails have more of these copy number aberrations than others, which is interesting. And also, patients that have NRAS mutations seem to be younger at diagnosis than others. And these are all interesting observations that are different from what is seen in cutaneous melanoma. So we're very interested in observing them further. Now, very briefly to tell you about consensus clustering. So this is RNA-seq data, and we can cluster by in an unsupervised manner by tumors that look similar to each other. This is the work of Esther Vasquez, another PhD student in our lab. And when you do that, then we see three groups. And these groups actually look different, but the one in the middle, which seems to have a better immune response, is actually associated with better prognostic characteristics like thinner tumors and just earlier stages. So we're investigating also whether those markers could be used as biomarkers to predict whether these tumors will progress or they are just good prognosis tumors overall. And finally, just to say that we can also profile the protein landscape of these tumors, and that's something that we're doing at the moment, which you're seeing here in this figure, with many different antibodies for immune cells. And at the moment, what we're doing is integrating the genomic, transcriptomic, and dysproteomic data of ALM tumors in collaboration with Julio Sáez at the University of Heidelberg to try and find correlations between RNA and protein and try to understand the immune landscape of these tumors. Okay, now I'd like to tell you a little bit about how we're building a collection of patient-derived models from Latin American patients. And this is very short. So what we do is we're in collaborating with Patricia Posic, who's a PI in Brazil. And what we do is we get patients from Brazil and Mexico. We get a surgical sample from the tumor, and then we inoculate it in the mouse, allow it to grow. And from there, we can study the evolution of the tumor, test different drugs, and also derive cell lines from there in order to characterize them by RNA-seq or exome sequencing, and finally find target genes and hopefully new drugs to treat these tumors. And at the moment, we have 96 patients, 64 from Brazil and 32 from Mexico, that have been inoculated in these mice. And I'm just going to tell you a little bit about the Brazilian cohort because it's more advanced. So for the Brazilian cohort, it looks similar to the Mexican cohort. So the ages, more common ages of diagnosis are between 60 and 80 years of age, stage 3 as well, some primaries, but also some local metastases, and most of the patients have had no treatment. And if we look at the success when we inoculate these tumors in the mice, then we see that about half of them do show growth. And this growth is associated with the thicker the tumor is, the better it grows. And also, if it's a primary, it grows not as efficiently if it's a metastasis. And we think that's because cells are more adapted now if it's a thicker tumor or if it's a metastasis, and therefore when we put them in mice, they grow better. Now, importantly, these PDX models recapitulate these histological characteristics, and they are capable of metastatic spread as well. Actually, about half of the tumors have shown metastasis in the mice, and in some cases to the same organs where the patients actually have shown some growth. So that's all to say that for this we have some ongoing work. We are characterizing them genomically. About 10% have mutations in BRAF of these models. We're also doing RNA seq of them to understand metastatic spread, for example, and what fusions are important. We're also monitoring the lungs, the brain, and the liver of these mice. With Patricia's team, we've been able to work with 6 sacral melanoma cell lines from these patients, and also try to model the anatomical locations, because at the moment what we do is inoculate these in the back of the mouse, but also it would be good if we could model better what the original location of this tumor was, which is palms of the hands, remember, soles of the feet. And finally, we have now in Mexico inoculated 7 samples, and 3 have already shown growth. So that's what we are working with. Okay, so now a summary of what I've told you today. We are working to establish a PDX collection from Latin American patients. Most inoculated tumors are stage 3, they're treatment naive and from older patients. We have inoculated about 40 PDX models, about half success rate. This PDX establishment is more successful when done for metastases and thicker tumors. These models recapitulate histological characteristics in patients, and half of these show metastatic potential. The ongoing work is focused on the genomic and transcriptomic characterization. And finally, some take-home message and acknowledgements. And the take-home message is that we hope that with this work we can contribute to the efforts to molecularly characterize acral melanoma, as well to close the gap in knowledge from understudied populations. And with that, I'd like to finish. I'd like to thank you for listening to this talk. Hopefully it was interesting to you, and also to acknowledge the lab. Here it's people, Christian, Patty, Steph, Fernanda, René, and Irving, which are the people that contributed to the work that you saw today, but also lots of collaborations with many different places around the world, specifically with the Sanger Institute, and our funders for their support always. And thank you. I hope you enjoyed it, and enjoy the rest of your conference. Thanks very much.
Greetings, everyone. Hope you are enjoying it. Greetings, everyone. Hope you are enjoying this conference and you have a nice time ahead and a very enriching learning experience. I am Dr. Gauri Mishra. I am presently a senior scientist working as the head of the molecular diagnostic lab, COVID kit testing lab, and sample receipt and report dispatch unit at the National Institute of Biologicals, Noida. Let me brief you about my institute. This is a autonomous institute under the Ministry of Health and Family Welfare. And we have been doing lot of work towards establishing the quality control testing of biologicals. So with my presentation, I am going to take you initially through the various aspects of quality control testing of biological, which NIB is doing. And thereafter, I shall be giving you a snapshot of what are the other research work that is being conducted at NIB and what are the opportunities for the students, researchers, and faculty at NIB Noida. So, NIB has a very vast infrastructure where mainly we have the diagnostic labs, therapeutic labs, There is a vaccine lab, and also there are support labs. Now, under the diagnostic labs purview, we have blood reagent laboratory, we have immunodiagnostic kit and molecular diagnostic lab. Also, we have biochemical kit lab, we have COVID kit testing lab, and COVID sample testing lab. Then, in the therapeutic labs category, we have blood products, recombinant products, enzymes and hormones, and monoclonal antibody lab. There is also a vaccine lab where different vaccines like BCG, COVID vaccines are being tested. And sterility lab and animal facility lab provides the support labs to all these labs for intra laboratory testing. Uh, the important thing is that all the labs and the biological products that are being tested at NIB Noida have an accreditation from NABL. And again, all these labs are CDL notified by the central government. NIB has many national and international linkages with various prestigious institutions. When we talk about the national linkages, we work hand in hand with ICMR, CDSO, various universities, blood centers, hospitals. Then students from various NIPERS, IT, and other reputed institutions come to NIB for hands-on training. Also, in the international section, we have a very close collaboration with NIPSI UK, EDQM France, WHO, NRL Australia, and there are different types of collaboration, not only in research but also in the proficiency testing that we do. Also, we provide hand holding to various countries like Bangladesh, South Korea, and Bhutan, where we help them develop and establish the quality control testing of various types of vaccines. Now, as you know that recently our country also globally COVID-19 pandemic was a very very big issue, and NIB has been instrumental in sharing the responsibility. And you will be happy to know that NIB has done almost more than during those pandemic times. NIB has been instrumental and it has done testing of more than 1.5 lakh COVID samples. Now, in our lab, basically what we do is the validation and batch testing of various COVID-19 diagnostic kits. We get different types of kits: COVID-19 RT-PCR kits, RT-LAMP kits, RNA extraction kits, and VTMs also. VTMs are viral transfer mediums, and we do the quality control evaluation of these. As a result, NIB ensures that only quality products are released not only in the Indian market but also only quality products are exported out of the country. And this is very very important because products which are not of good quality and products which are spurious. Let me explain. Spurious means, suppose you are taking a paracetamol. The label claim says it's a paracetamol, but then when you take the paracetamol for fever and it doesn't have a paracetamol, it has some other chemical in it, then we say that it is a spurious. It is not going to cause, it is not going to relieve the fever. So NIB is the only laboratories entire Southeast Asia which is presently doing the quality control regulation of the biologicals. Now, this is for the student's perspective. Let me take you to the basic definition. What do you understand by biologicals? So, generally, when we think about biologicals, they are basically derived from the living organisms or recombinant organisms by doing cultures of bacteria, yeast, plant, or animal cells, and includes diversified products like vaccines, growth factors, immune modulators, antibodies, and plus blood and plasma derived factors. Now, how it is different from the other medicines? Generally, what happens? The other medicines are either small molecule and they are either made synthetically or they are purified from plants. They are one biologicals market is the biggest one. Now, suppose if you have a product from a living organism and you identify it, you characterize it, and you purify it, you produce at a large scale. That is what many of the biologicals are, which are being used as for various therapeutic and diagnostic perspectives. So the different sources of biological products is they are either blood product derived or vaccines. They can be allergenic extracts, whole bloods, blood components, proteins, human tissues. They also involve various cellular and gene therapies and also xenotransplantation products. When you talk about biologicals, it is not only with the research purpose. Biological has a huge, enormous market. Indian biologicals is expected to rise around 12 US dollar billion by 2025. Can you imagine the market it holds? And there is a lot of scope for research and development in the field of the biological markets. This not only strengthens the Indian economy, but this growth, this prospective growth in biologicals is what the country is looking for in the form to support the Atmanirbhar Bharat. Now, when we see recently, you have seen during the pandemic times, India has been instrumental as a country. We not only produce the biologicals in the form of COVID vaccines, but also we provided 150 countries across the country, across the world. 150 countries were supplied with the COVID vaccines that have been produced in the India. And that was a really huge success for the India. Still, that trend which was capitalized during the pandemic times needs efforts, sustained efforts to grow in other fields of biologicals also. So when we talk NIB's role, actually it is at the interface. See, you understand the research, review, surveillance. So NIB handholds the industry, the researchers, the academia, and the regulators. It provides it is the R&D research and development based knowledge to the regulators to ensure that only the high efficacy and safety products are released in the Indian markets. Not only this, it ensures that all these biological testings complies to the regulatory guidelines. Now, as I have already mentioned previously, that biologicals should be of high quality, should be safe, and should be efficient. Each batch of a biological product should be tested for their efficacy to ensure that there is consistency, because you can have a batch to batch variation. Now, due to the differences in the nature and the regulations for different biologicals, they are tested either based on the guidelines that are present in various pharmacopoeias, or they are tested based on the manufacturer guidelines. There are different agencies worldwide which actually regulate these regulation for the biologicals. You must have heard of US FDA, or there is a medicine and healthcare product regulatory agency in UK. Similarly, you have therapeutic good administration in Australia, and India we have a central drug standard control organization which deals with the regulations, the application of license, the translating of the knowledge from the bench to the market takes place only when you have the proper licenses issued by the CDSO. These products, biologicals, when they are manufactured, and one subsequently they have to be tested at a large scale for their efficacy. When they are found of a standard quality, the same data manufacturers submit to the CDSO to grant the license for the manufacturer in the country. Now, there are two laboratories majorly in India, which we call as the testing laboratories. NIB, National Institute of Biologicals, tests almost 303 different types of biologicals. There are central drug laboratories like the one at Kasoli, which is into the testing of the vaccines majorly. Their main biological that they are testing is vaccines. However, NIB tests different types of biologicals. So See, presently this data is still 2022, but by as of date, we are testing 303 different types of biologicals, and all those biologicals are of very essential importance as far as healthcare sector is concerned. So the main role of NIB is to develop and validate standards for quality control testing for biologicals and immunobiological projects. Also, it helps in the development of linkages, collaborations with the national and international institutions, so that we are doing appropriate and we are progressing in the research and technological field to ensure that more quality biologicals are produced and tested. Also, we provide training facilities in quality control for different students, blood official professionals, faculties. I think we recently had a batch from your university, and we look forward to many more students coming to NIB and learn having a great learning experience. I can assure you NIB is the only institute that provides a hands-on experience to different latest techniques. Also, NIB generates assess the availability of qualified manpower. So the not only NIB is supporting the Atmanirbhar Bharat in its own way, but also supporting the Prime Minister skill development program, Pradhan Mantri Kaushal Vikas Yojana. Again, NIB is also coordinating the Hemovigilance program of India, where it ensures that there are no adverse reactions reported when the patients are transfused with different with bloods. Now, there are different challenges actually which are faced by the industry due to the complexity of the biologicals, their cost, their time from production to the market, and also due to the regulatory procedure. The major challenge I just mentioned was the batch to batch inconsistency, and also there is a lot of gap right now. See, when you produce a biological or simply when you do an experiment, you need a control for it. Similarly, for the biologicals, the presence of reference biological products is very important, and we do not have presently difference materials for all the biologicals. So when we understood these challenges, NIB in itself is trying to do its function to fulfill these gaps in the care market. We are trying to make the procedure, harmonize the procedure for development and testing of these biologicals. We are also trying to develop the reference materials so that the industry gets the support for development of different types of biologicals. Now, when we talk about the regulatory guidelines, there are different regulations. Drug and Cosmetic Act 1940. Similarly, there was Drug and Cosmetic Rules 1945. Recently, government is laying more emphasis on the many development and manufacturing of IVDs. Now, these IVDs are governed by the Medical Device Rules 2017. Similarly, for different types of biologicals and for different purposes, there are different forms in which regulatory guidelines are given to the, they apply in different forms for different purposes and get the necessary license from the CDSO. For different categories, now when there is a doubt that certain drug or certain biologicals is spurious or not functioning properly, then what happens? Drug inspectors seize that biological from the market and send it to NIB Noida. There are different scientists of the NIB Noida. I am also gazetted government analyst for testing of COVID-19 kits. Similarly, we have different government analysts and medical device testing officer who test the samples that are sent to us as a legal sample. Okay. Now, different products, blood grouping reagents, diagnostic kits for HIV, HBV, SCV, blood products, biochemical kits, glucose test strips, analyzers, different enzymes and hormones. There are different government analysts who have been notified as by the Gazette of India for testing the samples that come to us as a legal sample. Recently, I am happy to share that NIB received the best lab for the quality control regulation. We have been collaborating with various Asian countries, African countries for the testing of vaccines and other biologicals. Time to time, we also hold workshops for different participants. As I have already told you, NIB is very actively involved in the training the manpower, that is training different types of trainings. There are an entire training schedule present on the website. There is a dedicated procedure where you can apply for the trainings. As far as COVID-19 sample testing facility is concerned, till date we have tested almost 1,25,814 COVID suspected clinical samples at NIB. And during pandemic times, we have been 24 by 7 functional involved in performing this duty. NIB is one of the 24 centers initially which ICMR established, but later on, by the order of the Ministry of Health and Family Welfare, only NIB was the authorized testing lab which was given the responsibility of testing the different kinds of COVID-19 kits that my lab has been doing. Now, let me go to a research part which would be more interesting for the students. So, this is a snapshot of the different research work that my labs has been taking up. Recently, only we have signed it has been almost 1 year when we have signed a MOU with ACSIR, and we have initiated a PhD program for the students who qualify the national level test with fellowship. So through ACSIR, the students who clear the fellowship, different types of fellowship, whether it is DBT, DST or ACSIR, through ACSIR they apply for PhD at NIB. Now, with that, we initiated, and I am the coordinator, ACSIR coordinator from NIB for the PhD program that we have recently initiated. Also, we have ventured into research. That was one of the mandate of NIB. And there are different extramural research fundings that I have in my lab, under which we have tried to successfully perform some very good work. Hope you find this interesting. So one of the work that I initiated with was an in silico work previously, where there is a very important kinase. You must have heard of cell cycle regulation, G1, G2 phase, mitotic phase, and then you have a synthetic phase. So that phase is highly regulated. There are different type of kinases that actually function in different stages of these cell cycle. Now, there is an intricate network which actually regulates the transition from one phase to the another. Once a cell cycle regulating kinase is a mast which is a microtubule associated serine threonine like kinase. So this was a very recently, say only it has been 8 to 10 years when this kinase was discovered. So my lab actually has been instrumental in identifying. There were the inhibitors against this kinase. The best part was that we could identify the inhibitors from a natural source, and there are no significant inhibitors against this kinase in the market. The only one that was known at that time was the staurosporin, and we could find uh, better inhibitors than the reported one. So this was the in silico work that was published in 2018 in scientific reports. We then carried on this work, in silico work, and tried to do the validation in the cell lines, and we could establish that these identified inhibitors were giving a very good inhibitory potential. They had a very good inhibitory efficacy in the cell line. And this cell line based experimental validation was published in 2022 in the medical oncology from my group. Then This work opened many avenues. We then tried to understand what was the molecular mechanism of functioning of these inhibitors. To understand that, we tried to use the multidisciplinary approaches, not only in silico approaches, but also the biochemical approaches. The cell based studies were performed. Also, we did the next generation sequencing, and we could find that based on these, we could understand that these kinase, the inhibitors that were targeting this kinase were majorly affecting the apoptotic pathway. And one of the best inhibitor was flavopiridol, which was actually stopping the G1 transition of the cell cycle. It was causing the cell cycle arrest at the G1 phase. So this was a very good piece of work that has been recently published in December 2023 in a Life Sciences journal. There was one more breast cancer protein that we have been targeting simultaneously. It was known as the TET2. So this was a transcriptional factor, and we also identified the lead compounds that effectively inhibited this target. Presently, we are doing the cell-based studies validation of these targets. This work was published in 2023 in Briefings in Bioinformatics. So there are two aspects that my lab has been working on. One is, as I have shown you, a snapshot of the breast cancer biology. The other significant aspect, because I am also the head of the molecular diagnostic lab, where the major focus is on the infectious disease diagnostic testing like HIV, HPV, SCV. Another lab that I'm heading is a COVID-19 kit testing lab. So I have extramural funded project where we have been trying to understand how these kits can be made more efficient towards the ever evolving variants. As you know, the major challenge in any viral infection is the antigenic variation. Over a period of time, the virus gets evolved due to antigenic variations. And in case of SARS-CoV-2, this main antigenic variation is due to the changes in the spike protein. So we did a lot of study. We published some reviews, and also we studied what were the key signaling pathways that were responsible for the SARS-CoV-2 variants developed the severe outcomes. One is this. We have also performed the epidemiologic study from the Western Uttar Pradesh region, and we found that there were significant changes, nucleotide variations at specific positions in the spike protein that were responsible for the severe outcomes of the SARS-CoV-2 infection. And these particular variations, point mutations, were different in different strains. However, three escape variants were found in the S gene of the SARS-CoV-2, which potentially contributed to the immune evasion during the fourth, and particularly the fourth and the fifth waves in India. So this was a very important piece of our work on the SARS-CoV-2, which actually helped us to demarcate what was the variations in the current waves as compared to the previous waves as far as the molecular genetic makeup of the SARS-CoV-2 virus was there. We published this work recently in the Nature Scientific Reports. Again, not only this, my few of the PhD students that have recently joined, it is hardly 5 months back, they try to recapitulate all the developments that have been done in the breast cancer cell line research, and we have recently published a very good review. Manika and Supriya are my PhDs, current PhD students, supported by DST and DBT respectively, and we have published in a very good journal, International Journal of Cancers. Similar kind of work in we have performed where we did the inhibitor based screening of inhibitors, and then we did the validation of these inhibitors against an important protein and understanding how it affects the androgen receptor expression and what are the epigenetic modifications. This was a collaborative work with the Institute of Toxicology Research from Lucknow. Then there are work, these are snapshots of some of my previous work where I have contributed to the crystallography, and we have given some interesting insights into the Plasmodium falciparum HSP70 chaperone. There are 6 international books that has been published by me. The most recent one that will be coming, that has already been a soft book has been launched, and it is on the quality control and regulatory aspects for biologicals, regulations and best practices. The hard copy will be published in April. Also, I have published protocol handbook for cancer biology, which is actually very useful for the students. So if you want, you can consult that. And this actually give you different, what are the different SOPs, what are the different assays that you can perform in this field. The another very important book for the student perspective is the data processing handbook for complex biological data sources. I must say this is important because, see, when you read the, as far as the students are concerned, when they read the research papers, they understand the results, they understand the methodology. But then between this, there is a lot from bench to that research paper. There's a lot of data that gets generated, and it becomes very difficult for the students how to interpret that data or how to process that raw data to the final outcome to draw any conclusion. So this data process handbook for complex biological data sources has different techniques. You have mass spectroscopy, you have sequencing, you have fluorescence, circular dichroism. So if you are using any of those techniques, then how you can change that raw data to the, how you can process that data, how can you drive to a final outcome so as to give a particular biological interpretation to it. So this handbook, I think, will be very useful to the students. We have another book on omics, and introduction to biomolecular structure and biophysics was my first book, which is into one of the best sellers by the Springer. So presently, we have completed one ICMR extramural funded project on breast cancer. I have another ongoing project on the COVID-19 RT-PCR kits efficiency towards enhancing for different strains. And another CRISPR-based diagnostic development against dengue also we have completed. And there are other, recently we have got a very huge project on the development and validation of different in vitro diagnostics in collaboration with IIT Kanpur, IIT Delhi, IIT Roorkee, and also different medical colleges, Safdarjung. So I am the PI from the National Institute of Biologicals, and I have been collaborating with these institutes. And recently, only last week, we heard this that it got a sanction. So hope we will be able to contribute towards the IVD development and validation and handhold the industry, and we should be able to contribute towards the Atmanirbhar vision of the Honorable Prime Minister. So hope the NIB will not only be progressing day and night in the field of regulation, but this will certainly be a research mode backend development. So I welcome all the students. This is the patent that we have recently filed from NIB, and in the field of dengue detection of various dengue serotypes. Thank you all for your patient hearing. And in this email ID only, I have mentioned my email ID. If any one of you is interesting in any kind of research collaboration or any kind of support you feel NIB can give, so you are most welcome. I really thank the organizers for giving me this opportunity. Hope I had this previous commitment at in World Cancer Congress, so I couldn't come physically, but I hope in near future I will be coming physically to meet you all in person. You are most welcome to NIB, and if there is any support you require from NIB, we will be happy to host you. So there you must visit the NIB website, and if there is anything, you can reach out to us. Thank you so much. Thank you once again to the all the organizing team. You are doing a wonderful job, and I hope wish you all the best and wish you all a very learning experience. Thank you, everyone.
Hi everyone, I'll quickly go through my presentation about cerebral organoids and how we can use them in researching mostly psychiatric disorders. So, what are cerebral organoids? This is 3D self-organizing structures which are derived from human pluripotent stem cells, and the remarkable feature is that they can mimic very closely development of the human brain. They were first generated in 2013 by Madeleine Lancaster, and since have been reproduced successfully in many labs, including ours. So this is a slide showing the timeline of attempts to model the brain development in vitro, going from 2D models to 3D models. Starting with cerebral 3D models start with optical cups, then cerebral organoids, and later more defined models have been made, such as forebrain organoids, hindbrain organoids, and so on. Why do we want to use them? Mostly because of their human relevance. They are better than any existing animal models. They also model the developmental processes going on in the human brain, in developing human brain. Importantly, they allow experimental manipulation, such as drug screening and genetic modifications and so on. And also, very importantly, they can be made patient-specific from patient-derived pluripotent stem cells. Again, why do we use them? Because they do model the human brain very closely, as shown in this slide. Migrating neurons, maturing neurons, mature neurons, and so on. And those drawings have been made from stainings, where people stain for specific markers showing which cells are where and how they behave over time. So how do we make them? Most protocols are still based on the original protocol developed by Madeleine Lancaster, where you start with pluripotent stem cells and then produce embryoid bodies and then subject them to a variety of or sequence of growth factors to push them towards neurodevelopmental fate. We make them from human pluripotent stem cells. And what are human pluripotent stem cells? They are the cells that can differentiate into any cell of a human body. Historically, there are two types: human embryonic stem cells and induced pluripotent stem cells. And induced pluripotent stem cells are of our most interest because they can be made from a specific person, including a patient. We make them by taking a skin biopsy in our case, but other somatic cells can be used. We transform them with so-called Yamanaka factors, 4 transcription factors, which can convert them into pluripotent stem cells. Then we characterize the pluripotent stem cells we obtain, and we can use them for our differentiation experiments. This is an example of how the cells look. Again, in our case, we are using iPSC, induced pluripotent stem cells. We grow them in feeder-free conditions, and that's a picture of the cell line made in our lab in Odense. For now, we have made 5 lines from autistic patients and 6 control lines. The lines in green are already published, the rest will be published shortly. We also have over 80 lines of other psychiatric disorders such as ADHD, schizophrenia, bipolar, and so on from our collaborators. This is a slide showing how long it takes to start from the patient and produce a brain organoid specific for them. It's a total of more than half a year, 30 to 35 weeks. Of them, the last 3 months are making organoids, but the previous 3 months are making the line and characterizing it, so it's a lengthy process. Again, going back to the protocol, I just want to show how it goes on in our lab. So this is a picture published by Madeleine Lancaster in 2013, and this is what we do in our lab. On the right, you can see the cells that we grow, that we have derived ourselves and we grow in our lab, the embryoid bodies that we use for induction of neural fate, early organoids, where you can see the beautiful neural buds that form as soon as you put them into the induction media, and growing organoids which have cerebral tissue. Again, the picture on the right are the organoids growing in our lab right now. It's an experiment from February, but we have been doing it for 3-4 years now. What are the challenges in psychiatry, and why do we want to use brain organoids to study psychiatric disorder? Because diagnosis is often symptom-based and it can change over time, and we lack the understanding of molecular mechanism. Also, the drug selection is very random, and you try one drug and it doesn't work, you have to change to another drug. So if we could understand the molecular mechanisms better, we could perhaps personalize the drug administration, so it would be better for the patient, it would reduce their suffering, it would also reduce the burden on their families and society. So it is very important to understand molecular mechanisms of neurodevelopmental disorders, and brain organoids are a perfect model for that. So again, we want to use organoids because it's a human model, because we can manipulate them, because we can make them patient-specific, and we can compare them to organoids made from healthy controls. So we can try to figure out what's wrong with them, with the organoids that come from psychiatric patients, and hopefully develop better treatment for them. This is some earlier work from our lab, where organoids were produced from autistic patients and were subjected to proteomic studies, metabolomic study, and we have found 10 differentially expressed proteins which are, as I say, expressed differently in ASD organoids versus control. We also found the metabolism changes, and even the size was different. ASD organoids were growing faster than control organoids. That's another paper from our lab where we looked at oxidative stress from in autism-derived organoids, and unfortunately we could not see a difference. We could see that oxidative stress is growing with the age of organoid, but there was no significant difference between patient and controls. But we didn't give up. Perhaps we were looking in a slightly wrong way. We are going to try and look again using different disruption methods. It might be as simple as that. Another very recent publication from our lab, done in collaboration with Imperial College London, we looked at epigenetic modification in cerebral organoids derived from autistic patients, and we found that many methylation patterns are changed, and specifically Govt1 gene, which is important for the balance of excitatory and inhibitory neuron interaction in the brain, was specifically affected. What are we working on now? So there is a challenge recognized already by Madeleine Lancaster, where the efficiency of producing brain organoids is not very high. It begins with 60-80% at the neural bud formation stage, and only 30-80% will go on to become true brain organoids. We have also faced that even the size is very variable, as you can see. This is organoids produced from a single cell line in a single experiment. You can see that the size difference is as big as 3 to 5 times. And size difference might play a role. So we analyzed different sized organoids, as you can see here, and you can see that the smaller ones, or those that are closer to each other in size, are also closer to each other in protein content. So big organoids are not just more of the same, they are different. So if you want to make representative reproducible experiments, it's important that our organoids in a single experiment are as homogeneous as possible. So to that extent, we started to employ a 0-3D bioreactor, where the efficiency on this picture, you can see that the efficiency of neural bud formation is nearly 100%. Every single organoid you see in the picture, and that's reproduced in many experiments, is exhibiting neural buds. And also the size heterogeneity, compare the old picture to the new picture. They are a lot more homogeneous in the new when they are made in the new way, and also you can appreciate that we can make a lot more of them. So while in the old way we could make 20, maybe 30 per experiment, now we can make literally hundreds. And in that also allows us to pick up the better ones, the more homogeneous ones for our experiment, and it allows us to do technical replicates on them. So that's a rather important advancement. So yeah, this is the way we are making them in our lab now. We are able to produce high amounts of very homogeneous brain organoids, which we use further in our experiment. So now to the experiments we are on with. So we have done the experiment on bipolar organoids, or organoids derived from bipolar patients provided by our collaborator group. We've grown them and harvested them on 3 time points, 40, 70, and 100 days. And the 40 organoids got analyzed by proteomics and single-cell RNA sequencing. Very briefly, it's a recent results. We haven't analyzed them fully, but proteomics analysis have been able to find over 400 differentially expressed proteins. In single-cell RNA sequencing data, we found that organoid cells group into about 20 clusters, all neural-related. So they are brain organoids. They do contain mostly neural cells. And also, if you group the clusters by prevalence in human controls and bipolar patients, you can see that some of the clusters have more of the proteins from bipolar patients and others from healthy controls. Again, this is work in progress. Also, we have a list of genes that overexpressed or upregulated or downregulated in patients versus control, but we are analyzing the data, so that's about all I can tell for now. We also have similar experiments set up for adult ADHD, children ADHD, and a repeated experiment with autism patients. They've been grown and collected, and analysis is in progress now. So this is a recent experiment which we repeat with more ADHD children lines, and also already here you can see that ADHD organoids are bigger than control organoids, similar to autism organoids what we observed before. So, but this is an experiment in progress. Those organoids are growing back in Denmark as I talk to you. So hopefully we'll have more. So this is some work from other people showing what else they can do with organoids. This is a work done on a single ADHD patient compared to a single control. And the group was able to show that the cortical layers thickness is different between patient and control. But again, it was only done on a single person and a single control, so we don't really know whether it's representative of ADHD or whether it's person-to-person difference. We do know that person-to-person difference in organoids is significant. We are hoping to repeat it with more, so we have 4 controls and 4 patients for this particular disorder, so we are repeating it right now. Then you can also test drugs on organoids, just as I mentioned before. So here a group has created APSC cells and organoids from healthy controls and bipolar patients, some of whom are responding to lithium and some of them who do not respond to lithium. Lithium is one of the medications that can be used to treat bipolar disorder. And they show that, yes, indeed, lithium can rescue the phenotype of bipolar organoids, and responders respond to it better than non-responders. So now there is a way to go into molecular mechanisms and see what makes responders responders and non-responders non-responders. And also perhaps there is a way to predict whether people will respond to lithium or not. Yeah, you can also use organoids to study neurodegenerative disorders. But it's not as good as model as for neurodevelopmental disorders, because brain organoids represent a developing brain, not an aging brain. So maybe it's possible to make iPSC cells from older patients and see if it represents, for example, Alzheimer's disease better. But even now you can, for example, make genetic alteration to the genes known to be important for Alzheimer and test how the brain organoids will behave. So it is still a very useful model. something completely different. Brain organoids recently have been used in an electronic experiment, seeing if they can reproduce the activity of human brain for artificial intelligence and learning. And indeed, the hybrid computer, including lots of electronics and brain organoids, was able to learn the voice differences and improve the ability to learn. So it's still a very long shot, because brain organoids obviously are living things which are impossible to maintain in an electronic environment, but as a proof of concept, it works. What are the challenges in our work? Brain organoids unfortunately do not develop blood vessels, so as they grow and develop, they start to lack nutrition, and necrosis begin to develop from the inside of the organoids. So the outside of organoids still represent neural and brain tissues, while the inside is getting necrotic. So for now, there is no workaround. We have to analyze the outer layer, and again, heterogeneity in size and content, which we partly address with using the bioreactor we are using right now, but also heterogeneity between experiments might be significant, so we really have to control our experiments very rigorously. So yeah, that's a slide produced by Madeleine Lancaster, who says that you can do more specialized brain organoids, where the heterogeneity is less and homogeneity is better, but the complexity is smaller. So if you use spheroids or whole brain organoids, your complexity is not as high as in whole brain organoids, but your homogeneity is higher. So if you're asking a simpler question relating to a specific area of the brain, maybe you can use simpler system and get better reproducibility. If you work with complex system and want to ask complex questions, then you have to deal with more complex systems. Future uses of brain organoids and things I haven't mentioned is all possible questions you can ask: cell-cell interactions, electrophysiology of brain, screening of protein and RNA content, drug screening, inflammation, immunology, infections, and so on and so on. So sky is the limit of what you can use the brain organoids for. And finally, thank you, and this is the lab I'm working in, the Professor Tanya Sheljuk-Michel, my collaborator Sonia Hoffman, and former lab members who made the previous experiments and made the PSC cell lines, and our collaborators at Odense University Hospital who make proteomic and genomic analysis for us, and the collaborators in Switzerland and in Austria who produce us with iPSC cell from other diseases. Thank you, and I'll be happy to answer your questions.